Hi, Dr. Windisch from Sparks Pediatric and Adolescent Medicine again. Welcome to our video series for students. If you're a parent watching this, we're also more than happy to have you learning from us. And today this is really part of my ongoing Basic Sciences lecture series in medicine. And again, the topic has come up in discussions with one of my students about some basic sciences and how that relates to medicine and how we deal with that. So that's what we're going to talk about today. Please remember that these discussions are not intended to replace visits to your physician. If we can be of assistance to you, we're more than happy to see you here in our office. Our phone number, is 775-359-7111, and we'll get you in same day if you're having issues. But we really can't help you over the internet because that's just not good medical care. We're not interested in that layer. Okay, so what are we talking about today? Today, what we want to talk about is genetic probing, or how to study gene and gene product. So we're going to look at what, whoops, ah, there we go. We want to look at Western, Northern, and Southern blotting. And then some of their kind of precursor tests: polyacrylamide gel electrophoresis and agarose gel electrophoresis, and how that all works, and what is its various role in understanding what's going on with the gene. So we want to use an example of a gene and gene product. We can use any gene or gene product, and this can be used, and it doesn't even need to be human genome. It can be viral genome too. So you can be looking at the expression of uh, I don't know, P48 in HIV, or the expression of H1 in influenza, a certain influenza strain. But for the sake of making it really simple, we're going to look at keratin, hair. All right, and my question might be something like this. And again, for those of you who are true molecular biologists out there, please forgive me. This is a discussion of really of molecular biology as it applies to clinical medicine. So it is definitely kind of the Reader's Digest version. So, feel free to chime in the comments with details, but please don't crucify us, because this is not for molecular biologists. My molecular biology professors would have a heck of a lot more to say about this. So the first thing is, how do we study keratin? Well, the first thing you can do is you can look at the protein itself. All right, and what can you do? Well, you can look at its 3D structure either under an electron microscope, or you can crystallize it and do X-ray diffraction studies and get an idea of what its structure is. Okay, that's fascinating. We can now look at the size. So for example, I have a student with me who has really long blonde hair, and I have really short brown hair. Those of you who've seen my picture in other videos know what I look like. What is the difference between her keratin and mine? So we can roll out a gel, or pour out a gel of polyacrylamide, and polyacrylamide is just a chemical that you buy in a lab. Or let me make a better gel. That's really ugly. And you just pour out a sheet of the stuff. Okay, and then in the wells, you're going to put in your various samples of keratin that you've extracted. Maybe you want to compare keratin molecules across species. Maybe you want to compare keratin samples, across people. So let's actually do keratin across species, because that might be interesting as a general biologist. Or maybe somebody gave you some hair and you're working in a forensic lab, and your job is to say, you know, gee, is this hair from a human victim, or is this from an animal that attacked her or him? And if it was an animal, were they eaten by a bear or a rabbit? Okay, so we gotta get the species of animals. So the first thing we want to know is how big is the keratin molecule. So we will put our unknown in well number 1. And then maybe if I'm comparing my keratin molecules to those of my students, this would be me. So I'm going to put a male symbol here. And if we're comparing to my student, this will be a female because she's female. All right, and if we're looking at across species, maybe this is the unknown, and this is human, or this is a known sample of human, and this is rat, and this is dog. Okay, so these are our knowns, and we're going to use this to try to compare how does the size of this compare to the size of the knowns. All right, so we run it out with electricity, so we just run it in current, and we look and see how far the stuff travels. All right, so one comes here, and try to get my colors right. Another one is here, and another one sorry, another one is here, and where's my blue? This one runs out to be here. Whoops. That's supposed to be blue right there. Let's see if we can make that blue. There we go. Okay. Or better yet, we'll get that right. We want that even with that other red spot. Okay, so what we have here then is this tells me if we're looking at comparisons of my hair and my student's hair, that my molecule of keratin is not as long as my student's or big in terms of its aggregate size for whatever reason. Doesn't tell me what that reason is, just about the size. Or if we're looking at, and then I can run a known ladder of known keratin sizes here in another column, and it'll be a mixture of materials. And in this admixture that we put together, we know that this is 5 kilodaltons, and this is 10, and this is 20, and this is 25, and this is 30, and so on, or whatever kind of ladder, protein ladder you put together. And then based off that, I can now actually tell you how big the molecules are if I ran a ladder. Sometimes we're not interested in the ladder. Maybe I'm just interested in knowing species. So if we're looking at various species, I can tell you that this particular sample, my unknown, matches up with the rabbit sample, but it doesn't match up with human and with dogs, so we know this was a rabbit, just based off the size of the molecule, or at least it suggests it was a rabbit. Okay, so that is polyacrylamide gel electrophoresis. You can then take this polyacrylamide gel, okay, radio label it so that you make these radioactive by putting like a C13 tracer on there. Or actually what you're typically using is a tritium tracer, so an H3 tracer. Okay. And then you lay a piece of film across this and you let it sit in a refrigerator for a few days, and it's essentially going to just irradiate that film, and then you can take that film and develop it. That's called Western blotting. Okay, and by blotting it out with the film, it gives you an amplification, so something that's not this giant bright red dot like you see here, but actually more in another column over here, a dot like that where you're, oh, God, is that a dot? Am I hallucinating? Is that a flake on a fleck on my screen? Let me get what about that dot right there? There is a dot right there, by the way. I'll make it a little darker for you. Ooh, still can't see it. How about now? Now you can finally see it right there. But you're not sure, right? So we amplify it by making it radio labeled and laying it across a film. We call that Western blotting. So that allows you to look at the gene end product. Now, as a geneticist, that's not necessarily all that fascinating. So the next thing we may be interested in is the gene production itself, the RNA. So if you want to study RNA, you're going to do, and so maybe what we want to know is look at gene expression. How many copies of the gene are being turned out? So we might know what the RNA for a given gene, like the cystic fibrosis, actually looks like. And we know that in a specific mutation, the delta F508, it looks a certain way. And we can sit and just see how much RNA for delta F508 is being turned out in a given patient. Okay, or if we're looking at HIV, we may want to look at, again, how much of a gene product, how much what is it, 42, or in influenza, how much H1 is being produced in a given patient. So we take our RNA, and you got to be careful when you're working with RNA, and we load our unknown RNA here, and then we may load this one with known sizes of RNA. Okay, so these are our ladders, and we're going to again run it out. And in our unknown sample of RNA, and this isn't just delta F508, because this is a collection of all the RNA inside the human being that that we extracted it from. So we're going to get bands of RNA, big band here, little band here, medium-sized band here, all over the place in the patient. And you're going, yeah, which one's my delta F508 CFTR gene? How much product am I seeing? Well, that's all right, because here I'm going to run a known CFTR. So it's going to be blank, 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 and then the CFTR is going to light up right here. So that tells me that anything across this line is the same size as my CFTR. Okay, and then we're going to run a ladder of known mouse RNA strands. And in this ladder, we know we're supposed to have bands at the 16, 32, 132, 200, and 400 kilodalton range. So we can look and say, okay, this splotch right here is between the 200 and 400 kilodalton range. That's where we want to be looking. Okay. Now for Western blotting, or for northern blotting, we can take this one step further. So this is an agarose gel electrophoresis, and I've actually got this backward when I set my ladder up. The bigger numbers should be here. Alright, so 16,000 kilodalton, 320 kilodalton, 13 kilodalton, 2 kilodalton, 0.4 kilodalton. Because your larger samples of RNA, the bigger pieces won't go nearly as far through this goo as do the smaller pieces. Alright, so that allows us to look and just see, gee, are there pieces and how big are they? And maybe in my experimental system where we were doing gene therapy, we turned down the regulation of that mutant gene So it looks something like that instead of this big ugly band of whatever gene disease we're looking at. Okay. So we can see, gee, look, this band is twice as big over here as it is over here, so this patient's got twice as much RNA being turned out because we manipulated the gene somehow. We can amplify this one step further by radio labeling all of this, and we use H3 or tritium. Actually, it's H3. Okay. And so we tritium label usually like our thymidine. And then we can lay a piece of film over it, and we call that a northern blot. Or we can look specifically at the gene itself if we're doing mutation analysis. And we can run out on the same agarose, this time DNA, and we can do essentially all of the same tests we did up here. Okay, so if we're doing it with RNA, that's a northern. You can do it with DNA, that's a southern. We can also use it to isolate something. So let's say we're looking at a plasmid from where we were doing some cloning of a DNA strand. Okay, and this is the strand that we cloned. The rest of this is the plasmid P, whoops, the UC plasmid. Okay, PUC1. So we cleave out this, and what we're left with, we start with all the plasmid DNA. What we're left with is a mixture of two strands. Strand, oops, the strand we cloned in white, and then the rest of the native PUC1 strand, whoops, in red. Stop. We can take our mixture of DNA, which is just this syrupy mixture. We inject it, and it's a combination of stuff, so it'll be both of those. We inject it in a well here, and then we run it out. Okay, and then the big native remaining PUC1 will give us a blotch here, and then the small strand that we actually cloned will travel a lot further, and it'll give us a band here. We see this, we know that our cloning was successful, and that we were able to clone the gene, and maybe what I really want is I want this gene because I'm going to use it for other things. So now I can go in with a razor blade and essentially cut it out of the jello that it's in and clean it up with a process known as gene clean, and now I have myself a nice little vial of cloned genome that I can use. And this allows me to then study that gene or that portion of the gene in more detail with additional tests. So I hope this kind of helps make some sense out of some of what we're doing. It may actually confuse the issue a little insofar as we talk a lot about a lot of things because these tests are not used for any one thing. They're used for hundreds of other things. But by kind of giving you an idea of how we utilize them, it may help you to understand the tests, a little bit better, and just kind of make some sense out of it for you. So again, if we can be of assistance here, our office number is 359-7111, and we're happy to see you same day. If you're a student, we'd be happy to see you for a rotation. If we don't already have an affiliation with your university, that's easily arranged. We'll see you next time.